Good evening, everyone. Good evening, Say. Good evening, Pranjali. Good evening, Sahan. So, did any one of you faced any problems in the yesterday session? Was there any difficulty? Was it clear? Yesterday's class, was it clear? Okay, Hemant. Let me share my screen. So, before we continue with today's lesson, I want to clarify one small important point from yesterday's class. When we create a sequence record, so here we have created a sequence record in these two examples. In this example, we have created a sequence record, and here we have created a sequence record. So yesterday we learned that we create a sequence record because we want to store our sequence and we want to store the information related to that sequence. And that information is ID, name, description. So that is why we create a sequence record, right? So when we create a sequence record, we write attributes like ID, name, and description. So when we write these attributes like this, here we have mentioned sequence is equal to DNA, ID is equal to and then the ID, name is equal to and then the name, and description is equal to and then the description. So here we have specifically mentioned these attributes, but in this example we did not specifically mention these attributes. So if we do not write the attribute's name, then the Python assumes a fixed order, and that fixed order is sequence ID, then name, and then description. So if we do not specify the names, then we must follow this correct order. Firstly, we must add the sequence, then secondly, we must add the ID, then thirdly, we must add the name, and fourthly, we must add the description. But if we are specifically mentioning the sequence, we are specifically mentioning the ID, we are mentioning the name, and we are mentioning the description. So here in this case, the order does not matter. Okay, because here we are specifically mentioning everything. Okay, we can see example here. Let's comment this. So here we learned that the attributes order is like sequence, ID, name, description. So what if we write this description at the top and we write this sequence at the bottom? So here, as I said, that we are specifically mentioning the description, we are specifically mentioning the sequence. So here BioPython will not give us any error. So let's run this program. So here let's see in the output, we are seeing that these lines has printed us our output. Okay, so the point is if we are specifically mentioning it, we are mentioning the attributes, description, ID, name, sequence, then the order does not matter. But if we are not mentioning here in this example, we did not specifically mention the sequence, ID, name, description. So here in this example, we must follow this order. Is this point clear? Is this clear? Okay, very good. Okay, everyone, let's start with our today's lesson now. So, till now, we created the DNA sequence manually inside our code. Here, let's take the example. Again, let's see the example. See, here we created the DNA sequence manually, and here we created the protein sequence manually. So till now we were doing this. So, but in real bioinformatics, we never type sequences. Sequences come from machine, they come from database, they come from experiments. So, they come in the form of files. So, today we will learn how to read real biological sequence files using Biopython. So, first of all, let us understand what is a FASTA file. Just like I told you that sequences comes in the form of files. So FASTA file is one of them. Now what is a FASTA file? So a FASTA file is normally a simple text file which is used to store biological sequences like DNA sequence, RNA sequence or protein sequence. Here you can see one example. Here this is the example. So this is how usually a sequence is stored inside a FASTA file. As you can see that this is very simple and it is human readable also. So let's just understand what these two lines means. So a FASTA file usually starts with a greater than symbol. This is a greater than symbol. Whenever you see a greater than symbol in the starting of a file, that means that file is a FASTA file. So this is how you recognize a FASTA file. Now everything that comes after this greater than symbol is called a description. So everything after this greater than symbol is called a description. So here sequence underscore 1 means it is a description. So this description is about the sequence which is present at the bottom. So this is the actual DNA sequence and this is the description regarding this sequence, and the FASTA file starts with a greater than symbol. And see how easy it is. So FASTA file is very simple. There are only two parts. There is a header line. So this first line is also called a header line, and we also call it a description line because in this line we are seeing the description regarding that sequence. So this is the first part and the second part contains the sequence. Okay, so this is the reason why FASTA files are one of the most common formats in bioinformatics. So does anyone has any doubts regarding a FASTA file? Is it clear what is a FASTA file? Everyone, is it clear what is a FASTA file? Okay. Now let's understand from where we get a FASTA file. Just like we talked earlier, that sequences comes from experiment, they comes from databases. So where we should get this FASTA file? So a FASTA file comes from biological database. So let's open our Google. Now here in the search bar, we should type NCBI, as NCBI is a database. So NCBI stands for National Center for Biotechnology Information. Okay, let's open it. This is the official NCBI website. Now this is what NCBI looks like. So NCBI is a global biological database that stores here. It stores everything. It stores RNA. Here nucleotide stands for DNA. So NCBI stores DNA, RNA, here protein and many other things. So scientists from all over the world, they submit their data here. Okay, so today we will go to the nucleotide database. From here, from all databases, we will select the nucleotide. So here we have selected the nucleotide database. As you can see, there are many other databases present as well. So we are working with nucleotide database today. Now here in the search bar, we are going to search for a very simple gene. Okay, let's copy it. So here I'm searching a very simple gene. It is a mitochondrial gene. So here I have mentioned the specific name from which I want this mitochondrial gene. So it's Homo sapiens, means human. So COX1 is the mitochondrial gene, means it is a gene which is involved in the energy production inside mitochondria. Okay, let's click on the search button. So here we can see that NCBI gave us many types of nucleotide sequences. Here we can see many options, but for learning purpose, we will choose the first option. As this is a single gene and all others are entire genome, as you can see in the second option, it's written complete genome. In the third result, it is written complete genome. So these are complete genomes. That is why we are going to work with this single gene. So this is a COX1 single gene. So today we have to download a FASTA file. So here in this option, in this search result, as we can see, there are many options. So we have to click on this FASTA option. Let's click here. Now, as we can see, the NCBI has opened us the FASTA file of this gene. So here, as we have learned that how to read a FASTA file always starts with this greater than symbol, and anything after this greater than symbol is called a description. So this is our header line and this is our sequence line. So here we can see that in this header line there is greater than symbol, which means this is a FASTA file. Now after this greater than symbol you can see some numbers. Right now, what is this number? So this is called a ID. This is called sequence ID. Just like yesterday, we created a sequence record and we stored ID there, right? So each sequence has its own ID. So that is why this sequence ID is also called a unique identifier because it uniquely identifies each sequence. For example, if I copy the sequence, so I have copied it. Let's open NCBI again in the new tab. So here, let's open it. Now, let's search for nucleotide here. And now here, paste this sequence ID. So as we can see, I have pasted this sequence ID in this search bar. Let's click on the search button. So we can see here, Homo sapiens isolate mitochondrial cytochrome C oxidase. So this is the same gene which is opened here. Let's open the FASTA file. Click on the FASTA option. So we can see we have again opened the same gene because the sequence ID uniquely identifies each sequence. So each sequence has its own sequence ID. Okay, so this was about sequence ID. Now here this is the description that it is from Homo sapiens and it is the full form of COX1 gene. It is cytochrome C oxidase. So this is the description about this DNA sequence. Now we have to download this FASTA file. So to download this, here we will click on the send to option. Click here. We have to download the complete record. So it is already selected. We will choose the file option here. And here FASTA format is already selected. Now here we will click on the create file. So as we can see, the FASTA file has downloaded. Now let's open our file explorer. Let's go to the download section. So here we can see that the FASTA file has been downloaded. Now what we will do, we will cut this file from here. We will cut it and we will paste it in our Biopython folder. So this is the Biopython folder which we have currently opened inside VS Code. Here we can see this is the Biopython folder. Here Biopython course. Okay, and these are the files. So I will paste my file here. Now I have pasted this file here. So let's open our VS Code again. Now we can see that we are seeing the file here. So what we did, we just simply pasted the file in our folder here so that we can directly see this file in our VS Code. Okay, now what we will do, we will rename it. Let's rename it. So I am renaming it to cox1.fasta. So I have renamed my file. Let's open the VS Code again. So here we can see cox1.fasta. Okay, let's rename it again. Okay, so we have renamed it cox1.fasta, and we can clearly see this is the exactly same FASTA file which we saw in the NCBI. Okay, now that we have downloaded a FASTA file, we have clearly understood one thing, that sequences are stored in files, and a FASTA file may contain hundred or even thousands of sequences. So here, for learning purpose, we intentionally did not choose a very large DNA sequence. As we saw here, let's open the NCBI again. Here, let's go back. So we can see that we downloaded the FASTA file of this gene, and the length of this DNA sequence is 1542 base pairs, and the other length is 16,000. Here the length is around 16,000. Here the length is around 16,000 base pair. So the length of a DNA sequence inside a FASTA file can go up to even 50,000 base pair or 90,000 base pair. So those are very large DNA sequences. Now we can see what a 15,000 base pair length of DNA sequence looks like. Okay, so this might still, I can still copy it. Let's see, let's copy it. So I have copied it. Okay, let's rename this file. We will do it lesson9.py. Let's make a variable here, DNA, and save our DNA sequence here. Okay, there is some error here. So let's just see that this is a very large DNA sequence, and as we saw in the NCBI, that sequences can go up to 16,000 or even 90,000 or even more than 95,000. So there are many large DNA sequences. So to copy these DNA sequences and paste it here in our Python file, in our variable, it is very difficult and it is almost impractical. Okay, it's almost impractical. So what we need to do? We need to read these large files automatically. So that's why BioPython provides us a module called SequenceIO. So here BioPython provides us a module called SequenceIO. So here sequence IO, I and O stands for input and output, means we give the input and we receive our output. So what input we give? We give the input of our file, because this sequence IO module is specifically designed to read these FASTA files, to read these biological files. So now what is the first step? We have to import this sequence IO module inside our Python file. So for this, we will write from bio import sequence IO. So today we are importing a module. Today we are not importing a class. Till now we have imported a class. We have imported sequence class. We have imported sequence record class. So today we are importing a module. Let's hover over it. So we can see that this is a module. That's why we have written the code from bio import sequence IO. Let's see the example from the folder as well when we related. So here let's relate that this is the BioPython library. Now inside this BioPython library, these are the modules. So what code we will write? We will write from bio import sequence IO. Okay, so this is how we write a code. Now we have imported the sequence IO module. Now this sequence IO module will read our FASTA file. So what is our second step? To write a program to read this FASTA file. Now here let's create a variable. So here I am creating a variable record. So I have created a variable here record. Now we will use this module. Let's use this module here, write sequence IO. Now here in this module, there are some functions. So we are going to call one of that function, and we understood how to call a function. We add dot. So this is called dot operator, means inside this, means inside sequence IO. So inside the sequence IO, there is a function called read. And whenever we call a function, we add parentheses at the end of it. Right? Now inside this parenthesis, we will give our input. And what input we will give? We will give the input of our file because we are going to read this file. So here we will write our file name. What is our file name? Our file name is cox1.fasta. So here we have given our file name, and the second thing we will write the format of our file, that in which format the file is. So here our file is in the format of FASTA. So here we will write FASTA. So this is how we use a sequence IO module. We will use the sequence IO module like this. We will add dot. We will call the function read. We will add parenthesis. Now inside this parenthesis, we will give our input. We write our file name and we give the format of our file. So these are the two things that we give our input about. Okay. Now we have to read our file. So we have already used the sequence IO input, sequence IO module, and we have given our input here. We have given our file name. Now we have to read this file. So what is inside the file? There is sequence ID, there is description, there is sequence, all that we learned yesterday. Okay, yesterday we learned about sequence, ID, name and description. So that is what we are going to print. So let's write print the name of our variable record. Now inside this record, we have to print everything inside this record because all this line we have stored it inside this variable. So we will use our variable, add dot because we are going to print ID. As we saw that the ID is 0M986073.1. So this is the sequence ID. Okay. Now let's print description. So for that we will write record dot description and we will print the length of our sequence. So we also learned about length. So here we will write length. We will use the length function and we will give the name of our variable record. So inside this variable record, we have saved this, means we have saved the FASTA file and we have used the sequence IO module over it to read this FASTA file. So that means if we write print length of record, so here the length of this sequence will get printed. Okay, now let's run it. So here in the output, we are seeing that this line print record.id has printed us the ID. Now here, let's see in the FASTA file, this is the ID. And here the second line print record.description has printed us the whole description. Let's see again in the FASTA file. We learned that everything after the greater than symbol is called a description. So that's why in the output we are seeing the sequence ID as well as the whole description. Okay, and this line print length of record. So this has printed us the length of our sequence, which is 1542 base pair. So this is the same length that we saw in our NCBI. So here it is the same 1542 base pair. So this is how we read a FASTA file. Now I want to ask you one question. Yesterday we created a sequence record to save the sequence and to save ID, description, name and everything else. So here we did not created a sequence record, but still we printed ID, we printed description. So how I did it? Can anyone explain me any idea? Anything you know? Am I audible? Students, am I audible? Okay. Okay, Akshay, we will clear it. Very good, Ashwini. It's already in FASTA file. Yes, Amin, very good. Yes, Anurag, very well done. Okay, so Akshay, was that your question? Okay. Akshay, I think you joined late. We cleared this in the starting of our class that when we create a sequence record and when we specifically mention like ID is equal to then the ID, we specifically mention the name and then the name, and we specifically mention the description is equal to and then we write the description. So we are specifically mentioning the attributes inside the sequence record. So there the order does not matter. Okay, but if we are not specifically mentioning name is equal to, description is equal to, sequence is equal to, so for that we have to follow the order. Okay, Akshay, is it clear? Yes. Yes, everyone, very good. So here, what were we doing? We are using sequence IO module. So what this module is doing? This module is reading this FASTA file. So this module is automatically creating a sequence record inside it. Okay, so the sequence record is automatically being created. So that sequence record regarding this FASTA file already exists, and that's how we are able to print ID, we are able to print description. We can also print sequence as well. But as we can see that the sequence is very long, so it will get very lengthy in our output. So let's print the sequence as well. For that, we will write print. Then we will write the name of our variable record. We will add dot and we will write sequence. Okay, let's run it again. Now this time our output is going to be very lengthy. So here we can see we print the sequence as well. Okay, so that's why I didn't print the sequence. I only printed the length of the sequence. Is there any doubt how to read a FASTA file? Is there any doubt how to read a FASTA file? Is it clear? Okay, so if I give you a name of a gene, so are you able to download it from NCBI and are you able to read it? Like right now, if I give you a gene name to download from NCBI? Okay, very good everyone. Okay, so let me give you a gene name. Okay, so I'm writing in the chat box. Ayush, what are you not understanding? Okay, so are you able to understand? Okay, Samin, okay, let me share my screen again. So Samin, open your Google. Okay, here you have to type NCBI. So this is the official website of the NCBI, National Center for Biotechnology Information. Click here. Okay, now this is what NCBI looks like. Now this is the option all databases. So here you can find all type of databases here, but today we are working with DNA sequence. So that means we have to select nucleotide. So here you will select nucleotide and here you will write your gene name. For example, I searched for this gene name. So let's click on the search option. So these are all the searches. Here you can see the length of the sequence as well. Here the length is 1500, here the length is 16,000. So to download the FASTA file, you have to click here on the FASTA option. So click here, the NCBI will open you the FASTA file regarding this gene. Okay, so this is the FASTA file. Now to download it, you have to click here, send to option, choose the destination, you have to click on file option, then FASTA format is already selected, and then you will click on create file. Your FASTA file will get downloaded. Okay, it's easy. Let me know if you want me to repeat again. Riddhi, the format is FASTA. Okay, wait a minute, Abhay. Riddhi, the format is FASTA itself. Okay, Abhay, this is the code. Firstly, you have to import the SeqIO module by writing this code. Then you will create a variable in which you will store this line, how to read a FASTA file. So here you use the SeqIO module. Then you call the function which is inside the SeqIO module. This is the read function. Now here in the input, you will give the file name. So I renamed my file to cox1.fasta. As this is a FASTA file, so you must add .fasta in the end. As we are adding .py in the end, so this means this is a Python file. That's why .fasta means this is a FASTA file. Okay. And so here we write the name of our file, and here we give our the file format. So these are the two information that we give our input about. Okay, so this is the whole code. Okay, so I'm writing the gene name. Here you have to search for insulin homo sapiens. Everyone, you have to search for the gene insulin homo sapiens inside NCBI, download its FASTA file and read what we have learned today, how to read a FASTA file. Then you have to read that FASTA file and share your program in the group. Okay, insulin is a very small gene, so you should not face any problem. Any other questions? Vidasri, how to save in Biopython? Vidasri, have you downloaded the FASTA file? After you download the FASTA file, it should be available in your file explorer, right? From there, you have to copy and paste it in your folder which you have opened inside VS Code. So here inside my VS Code, I have opened this folder, Biopython Course, which is also in my desktop as well. So you have to save this file, the FASTA file, in your folder so that you can see the file here in the VS Code. So here, this is the program that you use to read the FASTA file. Riddhi, have you saved it inside the folder? As I am saying that you have to save the file, FASTA file inside your folder. So here my folder name is Biopython Course. Whatever your folder is, save your FASTA file inside your folder from your desktop. See here. So this is my desktop. This is my folder, Biopython Course. Here you have to save your FASTA file. Only after that, this file will be visible in the VS Code. Okay, because you are opening this folder inside VS Code. Riddhi and Vidasri, share your screenshot in the group. Everyone else, You have this gene name, insulin Homo sapiens. Search this gene name in NCBI, download its FASTA file and read that FASTA file and share your program in the group. Till then, Vidasri and Ridhi, share your file, share your screenshot. Okay, see, the problem is in the name of your file. Here on the left side, as you can see, the name of your file is sequence1.fasta. And here, what name are you storing in your input? What name are you giving? You are giving the name sequence.fasta. So fix this name. Either use that same name which you have stored your file, okay, or change or rename your file